Hey everybody. Okay, today we're going to be talking about food webs, and I'm pretty sure you probably talked about food webs. I know I introduced it in our last class period. I'm sure you probably talked about it in your lower level biology courses, but we're going to focus today on these coastal plant environments and the ecology of these coastal ecosystems. So I like this backdrop because this is now it's kind of a food web. It's really more of a food chain, but nonetheless, it was one of my favorite games growing up, and I think I was marginally good at it. All right, so we're still in this part of the course outline in the community ecology section, and my expectation is that you're going to be able to explain the roles of vascular plants in these coastal food webs, be able to describe the top down and bottom up controls of these food webs. You should understand stable isotopes, we're going to talk about a little bit today, and how they can help us better understand the interactions within these food webs. And then you should also be able to interpret data on consumptive and non-consumptive interactions across these trophic levels. So this is the first part of a two-part lecture series that we're going to hit over the next couple of classes. All right, so probably sometime in your life you've seen some version of this very oversimplified food web. Hopefully not at the college level, maybe K through 12. But generally you see these as very oversimplified in order to make them understandable at the layman level, right? The reality is they're far more complex. Practically inconceivable to some folks because of the level of complexity, right? Trying to trace all these things out. So this is more like probably what you saw in your freshman, sophomore level biology courses, where it kind of tracks through the trophic levels, right? And in many instances, you're actually calculating the amount of energy in order to determine generally what's happening within this food web. Okay, so and that's all fine and good. Um, if we use an aquatic food web as a example, however, um, the reality is that what's happening in these food webs is more complex, and it isn't necessarily equal between different members at the same trophic level. So we can utilize tools like stable isotopes that we're going to talk about today to better understand these relationships and what the relative importance of different organisms are within these very complex food webs. All right, so if you look at a food web or a partial food web here in the Galveston Bay system, so this would be fairly representative of most communities in, say, West Galveston Bay, right? And so you have your autotrophs, right? And then you have your low level, first level consumers. So these are going to be your herbivores, your detritivores. Then you might have your second level consumers and so on and so forth as you work your way up these trophic levels. But what is the relative magnitude of each of these interactions? And in the cases where you have one organism that might be feeding on several different things, what's the relative importance of those in terms of those interactions? All right. So one thing to understand is that ultimately all of these interactions depend on plants for nutrition, either directly or indirectly, right? Because plants are the ones that are converting the sunlight into biomass. And so that process then creates different types of biomass in terms of the carbon nitrogen ratio. So, for instance, in C4 plants, so a C4 plant is one that uses a specific type of photosynthesis called C4 photosynthesis. And so, in this instance, the first stable carbon compound that's produced during that process contains four carbon atoms. Okay. And so these plants tend to have a relatively high carbon to nitrogen level. And this is true of many marsh plants. So One of those plants is Spartina alterniflora. Now, we've talked about Spartina alterniflora. It is one of the prevalent plants in low marsh environments in the Galveston Bay system. And most Spartina, in terms of how it's consumed, it isn't through direct consumption. Now, that's not to say there aren't organisms that directly consume the leaves of the Spartina. There are a number of them, actually. But the proportion that's consumed is relatively low. So, like I said, there are some insects that will feed on these, will feed on the leaves of these plants directly. There are some mammals that will feed, and some birds also that will feed on either the plants or the directly or their seeds. So some notable ones are like West Indian manatees are one. Snow geese that actually utilize the seeds on their migration as a food source. But the amount of Spartina that's consumed after it has senesced and the leaves have sloughed off into the water during the winter, which supplies a huge amount of detritus into the sediments that are utilized by benthic organisms, including bacteria and fungi. Um, this is the most, in terms of sheer amount, is by far greater than the amount that is actually consumed directly on the live plants. We've also looked at mangroves. Here again, mangroves have a high carbon nitrogen level. They also have a high amount of tannins. So they have, like the Spartina, lower nutritive value, lower palatability. Again, this is true of most of these marsh plants. So that said, like with the Spartina, there are a number of plants that aren't, sorry, a number of organisms that are adapted either for direct herbivory or that are detritivores that consume the leaf litter as they fall down into the water. So crabs are critical components of these food webs. We looked at the importance of crabs in low marshes in the previous discussion. In this instance, crabs consume the propagules, they burrow in the sediments, which oxygenates the sediments, but also they drag leaves into these burrows where they break down. And they also consume both the live leaves and the leaf litter. And so this helps control decomposition and then the nutrient cycle, right? Those export rates. Seagrasses, right? So we've talked about seagrasses also as another layer in these marsh communities. So seagrasses here again have a relatively low food quality because of their high carbon to nitrogen ratio, but they do make a good substrate for algal growth. And they also produce a lot of detritus. So the common theme here is that plants in these coastal wetlands support a range of basal consumers. And this is through direct herbivory, through consumption of detritus, and through epiphyte growth, right? And that's true in all three of those subcommunities. All right, but what controls these communities? Is it primarily top-down forces, bottom-up forces, which is more important, right? Well, the fact is that in most cases, both forces interact, but the relative importance of each is really site and context specific. So if we look at an example where we have all marine plants, the studies typically show that the exclusion of herbivores is a little more important than the restriction of nutrients, more so when you look at seagrasses. However, this, as you're seeing here, where seagrasses are highly elevated, this is a relatively small N, right? So this study doesn't necessarily reflect what you might see across the range of seagrass communities globally, for instance. But again, generally, the enrichment effects are smaller than the herbivore effects, okay? But it is very site specific, right? Different communities, different players in the game, that can have an impact. I did in the notes leave you a reference to an article you can read that I think is pretty informative and might explain a little more of this. All right, so why do we care about this? Well, a lot of it comes back to adaptive management, right? We need to know how to properly design our management approaches in order to be able to, say, restore these biologic communities, maybe for their intrinsic ecologic values, maybe because of the secondary impacts that has on our anthropogenic impact interests. So maybe as these plant communities affect recreational or commercial fishing interests, for instance, which affects our significantly affects our regional economy here along the Gulf Coast. Or maybe it has to do with how these plant communities affect storm surge or any of those kind of things. These trophic relationships very much impact what these plant communities are going to look like and how they're going to function. But these trophic relationships, as I've alluded to, they're complex, and oftentimes you have both top-down and bottom-up interactions occurring. They aren't necessarily the same depending on what community you're talking about. And so ultimately the impacts, say for instance on seagrasses, it can be hard to tell what is the more important thing that's there, right? Again, we're talking about complex relationships. But stable isotopes can be a really valuable tool for us as we move through here. In fact, this is often the most powerful tool when we're looking at just how complex these relationships can be in terms of tracing the nature and magnitude of these individual relationships. So we've talked about stable isotopes before. In fact, we talked about carbon-13. So when we look at carbon specifically, we're going to look at nitrogen also in this particular context. So really, we're looking at the same element. It's still carbon. It has the same number of protons, which is the defining factor for what an element is, but yet is going to have a different number of neutrons. So the weight is going to be affected, right? That atomic mass is going to be affected. One isotope is relatively rare, the other is very common, okay? Again, the mass of the isotope affects the behavior of these isotopes. So in fact, they behave relatively similarly, but the speed of their reactions are different, right? And so this change is referred to as fractionation. So different plants have different types of internal reactions, and so they end up with different characteristic carbon-13 signatures, okay? And so you see this because the heavier isotope is discriminated against during the biogeochemical reactions. Each plant has a different fractionation rate, and so they have this unique signature. So you see this if, so I talked about C4 plants earlier. There's also C3 variety plants. There's a difference in terms of how much carbon-13, and specifically, we're talking about the ratio of carbon-13, kind of like we talked about with isotopes before. So you're comparing the ratio of carbon-13 to carbon-14 in the sample of the plant versus whatever its reference standard is, right? Same thing with nitrogen that we're going to look at. You see this in specific plant communities as you move your way up. And that can differ from the atmosphere, okay? So this really means there's some reality to the adage, you are what you eat, right? You guys remember Popeye, the spinach, I am what I am. So in any event, we use a couple of different isotopes when we're doing this work with these aquatic plant communities. Carbon-13 and nitrogen-15 is another. Again, the delta here is the same, right? We're looking at ratios of nitrogen-15 to nitrogen-14 in the plant tissue samples relative to a standard. Now, the standards In the case of nitrogen, it is the level of atmospheric nitrogen. In terms of carbon, it is typically a reference. The reference is Vienna PD belemnite. That's a mouthful. But belemnite is a fossilized organism that's found in the Beatty River in South Carolina. It is a typical reference standard. Um, with kind of the rule of thumb here is that the higher the level of nitrogen that you have, that ratio of nitrogen 15 to nitrogen 14 in the sample versus that in the standard, typically the higher the organism is in the food web. Okay. All right. So when we're looking at stable isotopes in these food web studies, um, the consumer level, so that the amount, the ratio of carbon 13 to carbon 12 in a tissue sample is going to be very similar to that of whatever it's consuming, right? What its diet is. It's similar with nitrogen 15, but there's a little bit of a bump that it gets. So there's a little bit of a variance there that is increased when we're looking at nitrogen. But in any event, it's very proportional to their dietary contribution. So what they're eating is very much going to affect how much of each of those isotopes is present in their tissues. So these are so I do some fisheries work, trophic level work in some of my research, and so when we are trapping fish out of these coastal marshes, what you find in shrimp, for instance, a grass shrimp is going to be different than what you might find in a croaker or a pipefish or any of these other organisms. Okay. All right. So what does this look like in practice? Well, all right. So if we look at an example here, when you look at stable isotope mixing, it provides a mechanism to evaluate contributions from different sources. So in its simplest state, right, if you've got an herbivore and it's just eating one thing, right, well, that relationship is pretty direct and it's pretty easy to understand what's happening there. But when you have a scenario where that organism is eating multiple things, which is far more common, this is much more often the case, then being able to identify these isotopes can, like carbon 13, for instance, can be really helpful in determining what the relative rate of consumption typically is. Now, typically you might think, well, it's just going to be this 50-50 ratio, and it may or it may not, because again, they may be eating more of one thing than another. So we can figure that out by looking at the ratio of these, the carbon that they consumed and how much is in their various types of food samples. All right, so let's use this example here. So if you look at a food web where you've got Thalassia, which is one of the types of seagrasses that we talked about previously. So this is our base organism. You have a lot of these other organisms in here. Grazers, so these are going to be things like shrimp and snails are going to have a relatively similar amount of carbon 13 and a slightly enriched amount of nitrogen 15, the ratios again, relative to the algae that they are consuming. Other plants are not going to be as important to these organisms. So the Thalassia itself is not going to be an important source of the stable isotopes because they're actually feeding on the epiphytes that are growing on the plants, right? So we can determine what they're feeding on, again, based on what these the sources of these stable isotopes are. Okay. That said, the amount of different linkages within these marsh food webs are typically far more complex. And so we're going to unpack more of that next time. So I'll look forward to picking up with you there.